Hello everyone. Today we have learned about the basic body plan of the flowering plants, part 6, in which we learned the double fertilization process of the angiospermic plants. Let us see one by one structure of the pollen grains. This is also called as the microspores, and these microspores are developed in the anthers. So this is the anther, this is connective, and this is filament. This is the whole structure is called as androceme. It is the male reproductive part of the plant. So at its maturity, this anthers which liberates many such a microspores, that is called as the pollen grains. It has exine and entine walls. Now let us see the further structures, that is the process of the microsporogenesis. Microsporogenesis is a particular process in which the synthesis of the microspore can be takes place, and it is released from this anthers. I just told you that this is a structure of the tapetum wall, and in which there is the microspore mother cell. This microspore mother cell undergo meiotic division and developing into the four kinds of the uh, cells. So out of which the one of the cell will be function, and this later on this pollen grain which germinates, that is sorry divide into pollen grains will be divided into two and forming into two nucleus, that is the generative nucleus and the vegetative nucleus. Later on, the generative nucleus which is divides to developing into two male gametes, and in this way the process of the microsporogenesis takes place. Now let us see how the development of the male gametophyte can be takes place, as shown in this particular figure, right from the figure A. So as shown in this figure A, here this is a pollen grain. So as I just told you, this is vegetative cell and this is a generative cell. So this is a germ pore which is present inside the outer and the inner, that is outer is a exine wall, inner is intine wall, and from which there is the germ pore will be developed. This generative cell which is divided and forming into these two male gametes as shown in this figure. Now this later on germinate and developing into the through this germ pore the pollen tube can be developed, and this from this generative nucleus the two male gametes are synthesized. Now let us see about the structure of the megaspore, that is the female gametophyte. Now this is the megaspore that we call it as the complete ovule. It is the female reproductive structure, and this is developed from this ovary, style, and the stigma. This is the structure of the gynoecium. So this ovule will be developed in this particular way. Now let us see how it will be developed. So there are the process of the megasporogenesis, and it's a development of the female gametophyte. So what is meant by megasporogenesis? Megasporogenesis is simply, we can say that it is a process in which the development of the megaspore, that is female gametophyte, can be takes place. So how it will be developed? As just we know that the megaspore mother cell, that is MMC, which is divides meiotically and forming into the two nuclei. So this four nuclei, out of which the only one will be functional, and this developing into the two nuclei. Later on it is tetrad structures, that is tetranucleated structure. Then this is octet stage, this is diad stage, this is the tetrad stage, and this is the octet stage. And this octet stage later on segregated this nuclei into two, which is that out of the four nuclei at the upper side, they developing into the two synergy cell and the egg, that developing into egg apparatus. One of the nucleus which is reaches towards the center, and from the lower four nuclei, out of these four, three will be developing into antipodal cells, 1 2 3 like this, and one will be reaches towards the center. And these two nuclei which is developing into centrally, they are having the secondary nucleus. So in this way, this is a complete structure that is known as the ovule or the female gametophyte can be developed. Now let us see the types of the pollen tube entry into the ovule, how these pollen tube will be enters into the ovule. So after germination of the pollen grain, pollen tube will be enters in the three way. There are the three ways of the entry of pollen tube. This is called porogamy, then chelazogamy and the mesogamy. Porogamy, through which the entry of pollen tube can be takes place through the micropilar end. Chelazogamy, that is the entry of pollen tube which is takes place through the chelazal end, and the mesogamy, that is the entry of the pollen tube which is takes place through the middle of the integument. So on the basis of that, there are the three kinds of the entry of the pollen tube into the ovule. Now how the process of double fertilization can be takes place. As I just told you that this is the male reproductive structure that is called as the androecium, and this is the female reproductive structure that is called as gynoecium. And this is the developmental stages of these pollen grains, that is microspore, and these are the developmental stages of the megaspore. So in this way, this pollen which is enters, it germinates through the on the stigma and developing into pollen tube and enters into the ovule, and they get fertilized. So in this way, the out of these two male gamete, one of the male gamete which is fuses with the egg cell, and remaining one male gamete which is fuses with the secondary nuclei. So how it will be developed? So this is the mechanism of the double fertilization. So this is the one of the male gamete fuses with the egg cell that developing into the zygote, and one of the male gamete which is fuses with the secondary nuclei, they developing into endospermic tissue. So zygote is a diploid and endosperm is a triploid in structure. Now this is the regarding the double fertilization mechanism. Thank you for watching. Thank you.